So hello everyone. So I will briefly recap what Ivana said, but also show our more or less in vitro data. So I'm Nikolina Mohović from University of Rijeka, and I'm working on the crosstalk of optineurin and TDP43 in ALS and FTD under the supervision of Associate Professor Ivana Mohović. So as you heard before, amyotrophic lateral sclerosis, or ALS, it's neurodegenerative disorder that affects upper and lower motor neurons. And because of the death of upper motor neurons, sclerosis of white matter lead to muscle weakness and amyotrophy. On the other hand, frontotemporal dementia, or FTD, affects neurons in frontal and temporal cortex, and leads to memory loss. So both of these diseases are characterized by high genetic heterogeneity, with some genes mutated only in ALS or FTD, but as you can see here, most of the genes are found mutated in both ALS and FTD, meaning that genetic overlap exists between these conditions. One of them is also optineurin that I will further explain a bit later. And before this, I want to pinpoint the main hallmarks of neurodegeneration, or in general, and also in this case, ALS and FTD. And first of them, it's TDP43 protein aggregation. So in healthy cells, TDP is predominantly present in the nucleus, while in the disease cells is depleted from the nucleus to the cytoplasm, where it forms phosphorylated and ubiquitinated protein aggregates. Another hallmark, as Ivana said, it's chronic neuroinflammation that is mainly driven by microglia, so that it's only immune-resident cell of the brain, but also some other cells like astrocytes. So there is the chicken-egg dilemma, what is happening first, but it is known that chronic neuroinflammation can trigger TDP43 protein aggregation and vice versa, and we are investigating the link between them. And we think the link is optineurin, and I will now try to explain you why. So to recap briefly what Ivana said, optineurin, it's non-enzymatic ubiquitin-binding adapter protein that it's directly implicated in inflammatory signaling by binding TBK1 and autophagy by binding LC32 and ubiquitinated cargo. Together with vesicle trafficking, these pathways are found altered in neurodegeneration and could lead to TDP43 protein aggregation and chronic neuroinflammation. So we are working on a mice model that has truncation of ubiquitin binding region. I will call it knock-in mice, and it's optineurin insufficiency mouse model because it has less truncated optineurin protein and less mRNA levels. But what is most important, it closely mimics mutations found in ALS patients. As you heard before, microglia is really important in neurodegeneration. So most of our study we did on microglia. First, we took BV2 microglia cell line and knockouted optineurin by CRISPR-Cas technology. As you can nicely see here, in our optineurin knockout cells, we have more TDP43 in basal state. Then we isolated neonatal primary microglia from our knock-in mouse model, and we saw the same phenotype. So in basal state, we have more TDP43 microglia. What's really important to say, said here, that this accumulation is post-translationally regulated because we did not see any change on mRNA levels in our cells. So as I mentioned before, optinurine has really important role in and several proposed role in microglia, in autophagy, sorry. So we wanted to test if the block of autophagy is the reason why we have accumulated TDP43. And we stimulate our cells with proteasome inhibitor, MG132, and autophagy inhibitor, bufalomycin A1. But as you can see here, we saw the a similar turnover of TDP43 protein in both wild type and knock-in cells. What is more important, we tested LC32 as a main marker of microglia, and you can see nicely here and also here that both in wild type cells and knock-in we have similar turnover and similar protein concentration, so LC32 protein concentration, meaning that increased TDP43 protein levels in a knock-in primary microglia for now could not be explained by autophagy block. I also mentioned before that it is shown that inflammation could trigger TDP43. And as optinurine has several proposed role in inflammatory signaling, we tested our primary microglia upon LPS, so it's lipopolysaccharide from gram-negative bacteria. And you can nicely see here that in wild-type cells, we have dose-dependent increase in TDP43 protein. But in our knock-in cells, TDP43 protein stayed at the same elevated state upon LPS as in basal conditions. We also checked for mRNA levels, but also could not see any difference. So here our hypothesis is that maybe our cells are chronically activated because upon LPS we did not see any additional accumulation of TDP43. So as I said you before, in healthy cells, TDP43 is mainly nuclear, while in the disease cells, TDP is depleted from the nucleus to the cytoplasm where it forms aggregates. So we wanted to see where our accumulated TDP43 is. And we stained the cells. You can see here. Do you see here? Yeah. So you will need to trust me. But basically, in wild-type cells, in basal state, TDP43 is mostly nuclear. Interestingly, and for us it was a bit surprising, in knock-in cells we have also nuclear TDP43, but the MFI is higher. So all the accumulated TDP in our cells were nuclear. What most surprised us, and it is in contrast to some researches before, that upon LPS, we did not see a nuclear depletion or protein aggregation. So accumulation of TDP happens only in the nucleus, in microglia. And somehow here we hit the dead end, so we went back to basics and did RNA-seq analysis because, as I said before, we were thinking that our cells are chronically activated, but surprisingly, in basal state, in our knockout cells, most inflammatory gene response were downregulated. Upon LPS, it was opposing, most genes were upregulated. So we think that optinurine works on several and different ways in inflammatory signaling. It has some kind of, and that our cells have some kind of altered immune response. So to conclude, I show you that our optinurine deficient or insufficient cells have increased TDP protein levels and that this change is post-translationally regulated and that autophagy is not the reason why we have more TDP43. I also show you that LPS increased TDP43 in wild-type cells, and that this increase is mostly nuclear, but fails to increase TDP43 in knock-in cells. So we somehow have already elevated state of TDP43 as in basal conditions. And I also show you that our preliminary RNA-seq data showed altered inflammatory gene response and suggesting that lack of functional optineurin could lead to chronically activated cells or cells with immunodeficiency that can consequently lead to increase in TDP-43 levels. I need to say that these RNA-seq data are just preliminary, and here we want to also test TDP splicing events and also downstream signaling of TDP-43. So this story, it's not over yet. With this, I want to thank everyone for listening. My associate professor, mentor Ivana Munitic, all of my colleagues, and also all of our fundings. And thank you for listening.